I just want to. Oh, no, no, no. Who started it?
I don't know.
It's Eric here.
Editor-in-Chief of MBoC, and also to introduce MBoC. This is the research journal of the ASCB. It's an important part of the ASCB community. So MBoC publishes research in all areas of cell biology and really at all scales as well, ranging from the molecular scale to the cellular scale to the tissue and whole organism scale, in all range of topics in the field of cell biology. So publishing in MBoC is a great way to communicate your science while also at the same time benefiting the ASCB community. And we publish a lot of great work in the journal, and we're here today to celebrate some of the best work published in the journal over the past year. In previous years, we've offered a single Paper of the Year award. This year, we've transitioned and expanded to offer what we're now calling the MBoC Early Career Paper Awards, and we offered multiple awards this year, and you'll hear from many of the awardees in today's session. So with that, I'll turn it over to Claire, who will introduce a little bit more about the award and also begin to introduce the speakers for today's session.
Good afternoon, everybody. Sorry, Matt, it's actually afternoon if you're on the East Coast.
Sorry.
Or Eastern time. Welcome. So my name is Claire Walzak. I'm at Indiana University School of Medicine in Bloomington, Indiana. And I am an editorial board member of MBoC, and I was on the board of reviewing editors before that for more years than I would like to count, and let you know my age. So one of the things we do is to select Paper of the Year, and just to give a little bit of insight in how we do it, the ASCB staff, when the manuscripts are reviewed, we take into account if trainees are lead authors on the study, and then we get a collection of all those papers published in the year, and the ASCB staff sends us a list of all of those papers, including the abstract and a link to the publication, and then we come up with a scoring system, and the entire editorial board looks for just gives their top 5, and then everybody there's a scoring system, and then the numbers fall out how they may. I think many of us approach it in similar ways. I always get the list. I do not look at the authors. I do not look at the institution, and I do not look at the lab from which the paper came, so that there is no bias like that. I simply read the abstract, and I think about the science in the broader cell biological field. So I'm always looking for papers in which whatever study, makes me think differently about a problem, or it's a long-standing struggle in this particular area, and then somebody had a sort of breakthrough finding or something that makes a significant impact. I tend to stay away from screening papers or my favorite protein type of papers, just because those tend to be, that's my personal bias that those, we publish plenty of those in my own lab. They're very important, and I love MBoC being a venue for that, but I'm always looking for this for a little bit of a distinction of something further. When Matt and I talked about this last week, Matt said he's the journal editor, right? He looks at citation, how many times it was downloaded or something. And I tend to ignore that, because sometimes somebody has a big family and then they send it out to grandma and 50 aunts. And if you're like my in-laws, you don't know how to use online or social media. And so my mother-in-law would probably download the same thing 750 times and still not actually have the article. So I just tend to ignore those metrics, but that's my personality. So congratulations to all of you. And then Matt, should we begin? And so I'm charged with introducing each of you, and then I realized as I just looked, I have your name, and then it says 7-minute talk and 3-minute question. So I'm hoping your first slide has the title of your paper because I actually don't have that handy. Our first speaker is Alejandro Melero. Did I say that correctly? Good. I tried to practice. And so please tell us about your work.
Should I then start? Okay. Let's see. Can you see my presentation? Fantastic. Okay. So good night from Spain. It's already 6 o'clock here. So I'm going to be talking about my work published in MBoC. It's a great honor to be here, by the way, today. So my work is entitled, or we better say, we studied the COPII vesicle formation by correlative light and electron microscopy with the aim of understanding the process of membrane remodeling from, with these vesicles. So let's start with what is COPII doing exactly, for those that don't remember. So can you see the mouse? The pointer? No? Yes? No. This always happens with Zoom. Okay, the mouse, yes, right?
Yes.
Fantastic. Okay, let's go that way. Okay, so this is the cytoplasm of any eukaryotic cell where we have several organelles and they are all interconnected through vesicular trafficking. All the vesicles share in common that they have to concentrate the cargo on the luminal side of the organelle, where we will have an outer cytosolic coat components that will recruit this cargo and induce membrane remodeling. COP2, similar to the other coat components, have different layers. We can subdivide the coat in an inner coat series of components and an outer coat series of components. The inner coat components are SAR1, which is a small GTPase, SEC23 and 24, that is a regulatory subunit, and very importantly, a cargo adapter subunit that has several paralogs. SEC24 has a paralog here called LST1, and here I exemplify the cargo binding with the P24 complex, which is a very common cargo in COP2 vesicles. Then we have the outer coat components, where we have an heterotetramer composed of long rods of SEC31 that are rigidified by this small protein called SEC13. Classically, it's been shown or thought that SEC13 and 31, because they are a cage with long rods supposed to be rigid, are the ones driving the curvature, while the inner coat components will be devoted to regulation and cargo recruitment to the vesicle. But more recent evidences were not exactly matching these conventional ideas. It was shown that the outer coat of SEC1331 may not be rigid enough to actually drive these budding events, while the inner coat components actually were shown to polymerize in very close tight arrays that suggest that could be actually participating in membrane budding, in membrane remodeling. So at the beginning of my project, I envisioned the idea of testing the different role of each layer of the coat by using classic yeast mutants that had been studied in other studies combined with genetics, biochemistry, or in vitro reconstitutions, recapitulate all these known mutations in yeast and observe them with electron tomography at high resolution. And to identify exactly where these events are in the yeast, I would use correlative light and electron microscopy. So going very briefly through the different mutants, they can be subdivided in inner coat component mutants and outer coat components. Starting with the outer coat, all of the deletions that can be done in the outer coat are lethal by themselves, but SEC13 can be actually deleted if it is combined with EMP24, which is part of this cargo receptor component called P24 complex. So it is thought that in the absence of the P24 complex, cargo is less abundant in vesicles, and that allows the formation of vesicles with a machinery that is more limited in membrane deformation. Then going to the inner coat components, I wanted to observe what happens in vesicles that are less loaded with cargo, and for that I use an EMP24 mutant. Then LST1, which is this cargo adapter, had been shown in vitro to produce larger vesicles. So it will be interesting to see whether this stands in vivo, in cellulo, better say. A combination of both mutations was also something interesting to do. And then SAR1, a mutation that is actually not a deletion, because SAR1 is essential for the process, but 2-point mutations that change the bulkiness of the amphipathic helix of SAR1, and it had been shown in vitro to have reduced capacity to generate curvature. So how is all of this working in vivo? Briefly, I will go through how we process the samples for correlative light and electron microscopy. First, we high pressure freeze our sample. This is a very amenable process for a big diversity of samples. Once they are cryo-immobilized, so this means they have been instantaneously fixed, if you like, we embed them in low acrylic resin that allows us to work at room temperature. And very importantly, also preserves the fluorescence in our samples. So if we have a GFP tag protein, we will be able to see it in the section for EM. So then once I have my sections, I will do the light microscopy together with some tetraspec fiducials that I will use for correlation. Subsequently, I will go to the electron microscope, acquire a tomogram, and then once I have the 2 sets of images, use these fiducials to correlate both images and identify precisely with about 100 to 200 nanometer precision the location of my GFP spot within the 3D electron tomogram that I generated, and seeing this structure with a nanoscale resolution. So it's a very powerful technique to study subcellular architecture. So I'm skipping through a lot of data for the sake of time. I'm going directly to some part, my favorite part of it, which is the analysis of the vesicle ultrastructure. Using a semi-automated segmentation analysis, we could retrieve the volume of these vesicles with high precision. It was automated. I didn't, the volumes reconstruct with a great precision out of the tomograms. Interestingly, all of the mutants, or not all, sorry, the SAR1 not, but most of the mutants actually impact the ultrastructure or the morphology of these vesicles. We had that the inner coat components were reducing the volume of our vesicles, while the outer coat component produced larger vesicles. Interestingly, when we decompose the vesicle in a long radius and a short radius, where we would have that the highest curvature is actually the short radius here, we observe something interesting. When manipulating or altering the outer coat component SEC13, the vesicles became elongated, while for the inner coat components, what we observed is that the vesicles became more curved in the short radius. This already, it's very eloquent about the different roles of the proteins. Is on the one hand showing that a coat with a limited rigidity in the outer coat can still make high curvature vesicles. The high curvature is still preserved, like in the wild type, only affected in the long radius, and it's suggesting that can be a problem actually with the fission of the vesicle. I'm not showing it for the sake of time, but in the SEC13 actually multiple buds accumulate at one single iris, while this is the opposite for most of the inner coat components, where there will be only one bud or even zero. I mean, obviously there are buds, if there are vesicles, there are buds, but they were less frequent to be seen. Then for the inner coat components, is suggesting that something is happening that we cannot foresee. It's not evident at first. So you have to think that when you delete LST1 now, most of the vesicles are formed exclusively with SEC24. LST1 had been shown to produce large vesicles in vitro. So this suggests that the different populations that we observe in wild type are actually mediated by predominant or diversity in the repertoire of coat, having that the smaller ones could be produced by only SEC24. Doing a similar analysis on the buds, this time it's not automated, this time I did it by hand, many budding events. We could observe something very nice, which is the trend of how the bud is generated. Here is all the different mutants. In the background in gray, you have the wild type budding profile. So if you see, please pay attention to the gray ones on the background, you see that they quickly go from shallow curvature, so this sort of stage, into a cup shape, which already has a high curvature. And then they progress in length, elongating the bud. And at some point, we don't see longer than, I think this was 80 nanometers or so. I mean, sometimes eventually you see one, but most frequently they will bud between 60 and 80 nanometers. They will pinch off. Now, if we observe the different mutants, a lot of cool stuff started to appear. My favorite is that when you reduce the cargo abundance in vesicles by deleting the P20, disassembling the P24 complex, the code immediately goes from shallow curvature very quickly to high curvature and elongated budding events, meaning that the code can quickly remodel the membrane because there is not opposition from the inner part of the vesicle. Then, when we look to the inner component SAR1, we saw that there's also skipping this part, essentially starting at high curvature. And this is contradictory, not contradictory, yes, contradictory, because it had been shown in vitro to not be able to produce high curvature, this protein. One thing that I'm not showing you, but I'm telling you, is that SAR1 actually was budding mostly from tubes. And again, this is an important feature of electron microscopy. I'm not just seeing the event that I want to see, but I'm seeing the context. And I could see that the budding events were mostly happening at very short, at very small radius tubes, telling you that when the SAR1 cannot contribute to curvature, the code components preferentially form vesicles where already there is high pre-existing curvature, adapting to the conditions. Then the outer code components mostly what we saw late stages of bud formation, not early stages shallow. That tells you that the progression from a shallow to a high curvature happened kind of normally, but then all of them stayed at the latest stage where they cannot pinch off. And this was something interesting, something new, because it's not clear how vesicle pinch-off scission happens in COP2. And this suggests that actually the outer code component may play a critical role in this part. So with this, and I don't know how fast I've been or not, but I'm going to the model to summarize all of our observations here. What we found is that, and this is similar to what has been shown to other codes, but partially here and there with in vitro genetics, biochemistry. What we saw with a correlative light and electron microscopy is that at the beginning you need to facilitate curvature formation because this is the very limited step. And for that, you will either go to tubulated areas or have the strength to do so. And you prefer low rigidity areas. We cannot see whether the areas are low rigidity or not. This is something we suppose because if they are already curved, that means that this is a deformable area. It's been shown by structural studies that the inner code can assemble into these arrays. And what we see is that actually limiting the code to a paralog that has been shown to do small vesicles enforces smaller radiuses as well in vesicle formation. So this suggests that the driver of this curvature of these radiuses could be actually the inner code that while forms these arrays, polymerizes the outer code. And what will be the role of the outer code if it is not driving curvature? It could be encapsulating all this curvature, allowing it to progress under the growing pressure within the vesicle. And while it grows in size and it grows in pressure inside, we will have now the opposite to the beginning. We will have an increase in membrane tension at the bud neck that will promote fission by disorganization of the membrane, either triggered by SAR1 or spontaneously. So the role of the outer code would be as an organizer of the inner code, organizer of the bud, and driver of membrane fission. And this was a feature that had not been suggested before in the literature, and we present evidence to support that. So this is the end of the story. I would like to thank many people, everyone in my lab during my time at the LMB, particularly my collaborators for other works like Natalia and Johan that were very important. Then the Kukulski lab, which I've been told that I am honorary member of the lab. I was I was the only one doing tomography in Liz Miller lab, while all these guys were tomographers. So I was always going down to chat with them, and they were critical and advancing my project. And then Jérôme Boulanger, who was critical for the light microscopy analysis of vesicles and ear exit sites. And I would like to thank the MBoC for this wonderful recognition. And thank you very much. It's an absolutely honor to be part of this. And all of you for your attention. I would gladly take any questions.
Thank you. It looks like we have a question from another one of our panelists. Louis, do you want to unmute and go?
Yeah. Can you hear me?
Yes.
Oh, awesome. Alejandro, that was an amazing, beautiful pictures. Yeah, and really jealous of those. So this is mostly ignorance, but when you have mutants of all of those COP2 components, do you affect fission the same rate, or like outer affects fission more than inner?
So that's a very good question, because it's something we noticed and we discussed that on the paper. So interestingly, when you produce vesicles with smaller radius, you increase the fission rate, and that makes sense, because a vesicle with a smaller radius will also be under more tension given a high curvature, and that would go in line with the idea of increased rate of spontaneous fission when you reach certain level of tension. While when larger vesicles or wild type level of vesicles were produced, this abundance of vesicles or potentially fission rate was less.
Oh,
We lost you.
yeah, we lost you at the end. Oh,
We still lost you.
can't hear you.
I do for that. I cannot do anything.
Oh,
Okay.
you're
Yeah.
good.
I think we need to move on because we're at 25 past the hour
Okay.
already. But thank you for sharing your very lovely work. Our next speaker in this webinar is Hongjun Song.
How do I?
Hello, everyone.
We need to switch sharing of screen.
Yes.
Yep.
That's it.
There,
Okay.
and now I think you can share.
All right.
Excellent, we can see it.
Okay. All right. First of all, I would like to thank everyone for coming to this exciting webinar. I'm Hongjun Song at Baylor College of Medicine at Houston, Texas. And today I will talk about how non-canonical Wnt ROR2 signaling pathway regulates cell matrix adhesion to prompt directional tumor cell invasion in breast cancer. Okay. As we know, breast cancer has become the most common cancer and the second leading cause of death in women. When it is localized, the 5-year survival rate is about 90%. However, once it becomes metastatic, the 5-year survival rate drops dramatically to less than 30%. So there is an urgent need to study metastatic breast cancer. So metastasis is a multi-step process involving local invasion, dissemination, tumor transient in circulation, and distant colonization. This process is dynamically regulated by many signaling pathways, among which Wnt signaling pathway is a key contributor. Extensive research have reviewed fundamental roles of canonical Wnt beta-catenin signaling in mammary gland development and breast cancer progression. However, the functional role of non-canonical Wnt pathway in tumor invasion and metastasis is still largely unknown. So this project focused on the non-canonical Wnt signaling in the breast cancer metastasis. In this study, we utilized a genetically modified P53 non-mouse model, which phenotypically and molecularly represent human breast cancer. Both the 2225L and the 2153L model that I will show you today belongs to the basal-like triple negative breast cancer. In these breast cancer models, when we knock down ROR2 with shRNAs and culture them as organoids in collagen, we can see that ROR2 depleted tumors are significantly more invasive than their than the control group. Our quantification confirmed that both the number of invading protrusions and average distance of invasion increase when ROR2 is knocked down. To understand the molecular mechanism underlying this tumor invasion, we conduct RNA sequencing on these tumor organoids, and the gene ontology turn enrichment analysis shows significantly altered collagen organization. With collagen hybridizing peptide staining, we observe elevated collagen abundance in the ROR2 depleted tumors. Using 2-photon-second harmonic microscopy, we captured the realignment of collagen fibers in the ROR2 depleted tumor organoids. These results indicate that the loss of ROR2 increase collagen reorganization and facilitate invasion. Collagen is a major component of extracellular matrix, and the integrin family are critical transmembrane receptors responsible for cellular extracellular matrix interaction, which regulates cell adhesion and migration. Interestingly, we find that the integrin signaling is significantly upregulated in our gene ontology turn enrichment analysis. Further immunostaining and biochemistry studies confirm that alpha-5 integrin and its ligand fibronectin is increased both in vitro and in vivo in our models. Focal adhesion complexes are direct downstream effectors of integrin signaling and serve to modulate cytoskeleton structures. Our organoids revealed an elevation of phosphorylated focal adhesion kinase in vitro. Meanwhile, our tumor model showed an elevation and the redistribution of phosphorylated focal adhesion kinase in the tumor body. Further study in the actin cytoskeleton dynamics revealed increased F to G actin ratio and reorganized F actin stress fibers in the ROR2 deficient tumors. Atomic force microscopy captured increased cell stiffness in the ROR2 depleted tumor cells, confirming our finding that the stress fiber is changed. Downstream signaling pathway analysis revealed decreased ROC1, ROA, and phosphorylated myosin light chain 2. In addition, the F-actin disassembling protein cophilin is also reduced in the ROR2 depleted tumors. So in summary, our manuscript at Molecular Biology of the Cell shows that the loss of ROR2 will promote the ECM remodeling and increase fibronectin integrin signaling, which will promote tumor invasion and dissemination. And with inhibitors of the integrin signaling and FAC, we show that this signaling is required for tumor invasion. Our ongoing project is to study how the ROR2 and non-canonical wing signaling can affect the CTC transit in breast cancer, as well as the ECM landscape and tumor cell survival in metastasis. At last, I would like to thank the Roddy Lab and Rosen Lab at Baylor College of Medicine. I also would like to thank Molecular Biology of the Cell for this great opportunity to share my research. If you're interested, please refer to our paper for more details. I'm also very happy to answer any questions you may have. And at last, I would like to thank all the funding sources that make this research possible. Thank you.
Thank you. That was an amazing amount of data in a very concise talk. Great job. Do we have any questions? I can ask one. So what you said, one of your next steps was to look at the circulating tumor, right? The CTC. So, yes. So you just think that it's basically an escape mechanism because of the increased migration. For a minute, I thought you were looking at circulating tumor DNA, and then I was really confused about how that could be increased. But you said it was increased invasiveness, but I think I missed what was the data for the increased dissemination, the escape that you would find it?
Yes, it's a great question because in this study we find that the cell adhesion is changed upon the ROR2 loss, and in the CT in the tumor transit in the circulation, it's very important for those tumor cells to interact with other tumor cells and also immune cells and also endothelial cells to have a successful transit from the primary side to the secondary side. So we think this molecular mechanism may also play a role to decide how successful these cells can leave the primary side and colonize the secondary side. Meanwhile, it has also been shown that the cell adhesion and ECM is critical for the tumor cell survival in the circulation. That's why I think the CTC may be the ROR2 signaling may be critical in the CTC survival and transit in this case.
Excellent, thank you. Are there any other questions? I'm not sure. If a panelist, if an attendee raises their hand, will I see it on the panelist screen? Sorry, because we
Not
can't see
sure,
the
but
faces. Yeah,
I-
exactly.
I think though that if a attendee has a question, they could put it in the Q&A
Right.
box or the chat, or they can attempt to raise their hand. There's multiple ways they can
Yes.
ask.
I mean, I think they can raise their hand because I have a thing that says lower all attendees' hands. So,
I think so too.
okay. Are there any other questions? Okay, if not, thank you. We can always come back at the end. That was lovely. So our next speaker is, forgive if I brutalize your French. That's me, not you.
Not French, this is Spanish.
Okay, see, even more of a thing. So Louie Ponce. How bad?
Oh, it's good. It's good. It's
Okay.
good. Don't worry about it.
Is our next speaker. Thank you.
I have a disabled screen sharing. So
You're still not able to share your screen?
I am not. I mean, because this is disabled.
Luis, can you share the PDF with me? I'll share the screen. How about go next one, Claire?
Yeah, nice one. Yeah.
Yeah,
Okay.
for Luis, send that to me. Thank you.
Okay, so we're going to go out of order then. So Ruben, I hope you're ready. So then our next speaker would be Ruben Phillip. And hopefully they can share their screen.
Can you hear me?
I can hear you, but we don't see your screen yet.
Oh, just give me a second.
I see a beautiful wave.
Okay, awesome. Can you see that?
I see beautiful microtubules.
Awesome, awesome. All right. Okay, so I can start?
Yes, please.
Okay. All right. So to begin, I'd first like to thank the organizers as well as ASCB and MBoC for this recognition. Today, my talk will revolve around the study we published in MBoC last year. I'll show you a few key pieces that I've pared down of data from this study, looking at how terminally differentiated osteoclasts organize centrosome into clusters for microtubule organization and bone resorption. But before getting into it, I'd like to, let me just get my laser pointer going. Do you guys see that? Can you guys see my laser pointer?
Yes.
Sorry. Okay. Before getting into it, I'd like to give you some background about these cells and where they act. When thinking about your skeleton, bones are commonly perceived as rigid structures whose sole purpose is meant to hold your body up. But in actuality, it's an extremely dynamic organ that's constantly undergoing remodeling. This remodeling process is carried about by two primary cells, the osteoblasts, the bone makers, and the osteoclasts, the bone eaters. And together they coordinate to form a homeostasis of bone turnover that ultimately maintains strength and integrity of your bone. So conducting research on these cells is crucial because any imbalance in this homeostasis can result in severe bone-related diseases, such as when it's shifted to the left here, you have the overproduction of bone. It can lead to osteopetrosis, where you have abnormally dense bones. And what is probably more pressing and prevalent within our population is the shift towards increased degradation, which can predispose individuals to conditions like osteoporosis or brittle bones. So focusing on the osteoclasts then, they're an extremely unique cell type to study due to how they differentiate and how they function. They originate from hematopoietic stem cells in your bone marrow. These stem cells then differentiate into a macrophage lineage, after which, upon reception of the rank ligand or rank L, the progenitors begin to rapidly fuse with one another to form a multinucleated body, which then can polarize and then resorb bone. So looking closer at its functional state, I hope you can appreciate how specialized and complex they are, but I'll highlight a few key structures that are formed during resorption. So once targeted to a site of bone resorption, they'll form a sealing zone made up of actin to isolate this resorbed area. The ruffle border then is formed to pump various proteases and ions to degrade the calcified bone. And then the resorbed material is trafficked across the cell to the secretory domain for release. So many of these processes require large cytoskeletal changes and coordination. And being a microtubule lab, we initially asked where the microtubules are being nucleated from, where are they coming from? So normally the centrosome fulfills this role in dividing cells as the primary microtubule organizing center, or MTOC, where normal cells, it plays a variety of roles, including maintaining intracellular structure, trafficking, directing migration, forming cilia, and being crucial for the formation of mitotic spindle during division. But osteoclasts are terminally differentiated, so they don't divide. So we began looking into studies that were inquiring about the MTOCs within these specialized cells and came across a paper in 2002. And just to summarize their conclusions, it was that they found little to no centrosomal MTOCs in these osteoclasts, these big multinucleated cells. They hypothesized that the centrosome would inactivate upon fusion and the microtubules would nucleate from other sites in the cell, proposing a similar mechanism to what occurs in myotubes, another cellular fusion model that holds their microtubules along the parallel arrays along their nuclei. So we initially took issue with this model because there's one key difference between these cell types. Osteoclasts are much more active and they're much more motile. And just here's a movie of them degrading a bone mimic. And you can see they're moving a lot. Their actin is constantly remodeling. And you can see the resorption pits they leave behind as they move across. So to find out what's really going on here, we initially began with staining experiments where we stained for nuclei to mark them. We stained for the actin ring to see the polarization and the sealing zone formation. And finally, we stained them with microtubules. And what's really interesting, an interesting thing we saw right off the bat is these radial arrays. And typically radial arrays are indicative of centrosomes. We further then marked the centrosomes with an antibody, and what we saw was that they were actually organizing into large clusters, and these clusters correlate with the amount of nuclei within the cell, the final cell total. So after quantifying the centrosomes across the polarized state as well as the non-polarized state, we saw that centrosomes generally persisted after fusion and forming the final mature osteoclasts, as well as adopted this unique clustering form. We also performed movies to see if this is true and how soon they clustered after differentiation. So here, just keep your eye out for cell 1 and cell 2. And then we have centrosomes labeled in green here. I'm going to play the movie. We'll see it as cell 1 fuses with cell 2 that the centrosomes kind of come and kiss. almost immediately after fusion. So knowing that they cluster, we wanted to see how important these clusters were to their actual function, bone resorption. We actually came across only one model where centrosome quote unquote clustering exists, and this is actually in cancer cells. Cancer cells often adopt extra centrosomes and have to keep them in clustered orientations in order to not compromise their cell division. So we came across this study where they used a various amount of antimitotic and -N motors inhibitors and saw that they could disperse these clusters within interface. So we tried to see if we could replicate these results in osteoclasts, and here's just an image of the strongest inhibitor, Phillip. So here, before the treatment, we see a tight cluster of centrosomes, as indicated in that right there. And then after using the drug, we see them all dispersed across the cell. And quantifying this with this definition of a lone centrosome being more than 1 micrometer away from any other centrosome, we saw that clusters as a whole in the presence of these drugs were drastically reduced, and the distribution of lone centrosomes increased drastically. So then we sought to figure out, do these affect function? And the first things we looked at were the size of their resorption rings. So we, again, treated these with the 3 drugs that we intended to try, and then we had these osteoclasts plated on a bone mimetic cover slip. And what we can see is we see these large resorption rings in the untreated control, but then upon treatment with these declustering drugs, they tended to not form large actin rings. And as a result of not forming these large actin rings, the overall total resorbed area of the bone mimetic was drastically reduced, as we can see here. So just to quickly summarize then, what we found was that in the specialized sen, osteoclasts maintain their centrosomes after differentiation, and cluster them in order to aid with actin ring formation as well as bone resorption. As far as our lab's future inquiry, it will lie in nailing down the centrosome clustering mechanism, as well as exploring what centrosomes are doing during osteoclast fusion. And finally, just as a general expansion to this project, we're curious about how osteoclasts are coordinating all their other organelles within these super specialized cells. So with that, I'd like to just thank my supervisor, Dr. Harrison, for the support on this project, as well as the members of the Harrison Lab, particularly Cara Fiorino, who's been my partner on this project. And I'd like to thank you all for listening.
Thank you. I will have to keep the questions short, but what I would also like to propose is that we can do like a short question now, and then for anyone who's interested in more detail, I hope we are not cut off at 1 if we want to, because I have a bunch of comments since I love C-terminal kinesis. So I see the hand up from Hongjun.
Yes, beautiful work. I have a quick question. So you did show 2 breast cancer cell lines showing the clusters. Do you know why they have those clusters, and do all breast cancer cell lines have that?
So there's a global review and a lot of clinical studies of histology slices from primary patients, and it seems like a lot of cancer cells, especially those that are undergo EMT, they adopt extra centrosomes for some reason. And it's like a chicken and egg scenario as to whether the centrosome duplication pathway just starts generating extra centrosomes or a mutation induces that. But generally, typically tumors and especially metastatic cancer cells, especially a lot of breast cancer cells seem to have a high amount of centrosome amplification or extra centrosomes. And when they have this, no matter the cell, they often hold them tightly together, all those extra centrosomes, because when you have the centrosomes going all willy-nilly, instead of forming a nice bipolar spindle to divide, you're going to form tripolar or multipolar spindle, which is obviously not great when you're trying to segregate chromosomes.
Thank you.
Yeah.
Okay. Thank you. And as I said, we can stay on at the end. So with that, let's go on to our final speaker. We are back to Louie, who now has a screen to share. So if you'd like to get started.
Did it work? Can I share?
I don't know. I see the paper. I don't know if it's yours or.
Fantastic. That was okay. That was really cool. All right. Thank you so much for the award. It was really cool, and for allowing me to present my data. This has been a really nice meeting, learning a lot of different things. So I'm going to present this work that I did at my time as at NIH as a fellow. Now, as you can see here, I'm a Buckeye, but I'm really proud of what we did there at the NIH. So this is basically understanding how the Parkinson disease protein LOR2 plays a role in lysosomal tubulation and the role of the endoplasmic reticulum in fission. So in this project, my main focus was to understand how this large protein called leucine-rich repeat kinase 2 and its biology, and sort of understand the biology of the protein. Here is the cryo-EM representation of the structure of the protein. What is important is that it is a setting-threatening kinase. And the reason why I am interested in LOR2, in short, is that LOR2 has been linked to different human disorders. So it's very well known that LOR2 affects and increases the risk to develop neurodegeneration, especially Parkinson's disease and progressive supranuclear palsy. But also it is very well known that LOR2 can also affect and is linked to different diseases in the periphery, immune diseases like Crohn's disease, leprosy, or infectious disease. So basically understanding the biology of LOR2 is very relevant to understanding different human disorders. The problem, though, is that the role of LOR2 in the cell is a pretty unknown and difficult matter to understand. And therefore, if we don't understand why a protein causes a disease, it's really hard to find proper therapeutic targets. What was known in 2016 is that LOR2 can phosphorylate 14 RAB GTPases. And this is very relevant at the time because as RABs are main actors in membrane trafficking, this sort of put LOR2 indirectly affecting something among membrane trafficking, organelle dynamics, and so forth. So among the potential options, I focus on lysosomes, here staying in orange. And obviously lysosomes are extremely relevant organelles and are crucial in all of the diseases that LOR2 is linked to. But also, and I'm stealing this sentence from Juan Bonifacino, those are lysosomes. And that means that lysosomes are not only relevant for cargo clearance, but they promote different functions in the cell, and all of them are really crucial under stress conditions to preserve cellular homeostasis. And so the whole point of my project, my time at the NIH, was to understand whether LOR2 plays a role in lysosomal biology. And so what we did in the paper previously to the MBoC one was to try to understand what LOR2 could do on lysosomes. And what we found is that LOR2 is normal, as you could see here in cyan, is in the cytosol. And when you treat cells with a lysosomal disruptor, in this case, LOMI, that breaks the lysosomal membrane, what you will hopefully see in this movie is that LOR2 starts to be recruited in punctate structures in long periods of time. And those punctate structures are actually lysosomes, as you could see for the colocalization with LAM1, which is a lysosomal membrane marker. So what we saw is that actually on lysosomes, through its kinase activity, LOR2 recruits a protein called GIP4, which is a motor adapter protein. And GIP4 forms these beautiful tubular structures that come out of the lysosome. And what we saw is that if you track them over time, those tubules can actually get sorted into vesicles. And what we realized is that we just stumbled into a new cellular process that we call LITO, for lysosomal tubulation and sorting, driven by LOR2. What we've seen about these vesicles is that they can actually interact with healthy lysosomes. And what we think, and we're working on it right now, what we think is that this is a way to deliver undegraded cargo or toxic materials. The lipids could be protein aggregates from dysfunctional lysosomes to healthy lysosomes in order to maintain cellular homeostasis. So basically, this is a model that we found on the Science Advances paper, and it's very simplistic. Now we know it's a little more complicated than that. It's based on LOR2 recruiting its substrate for soraptan, and that's triggering GIP4 recruitment, and some motors we're looking at whether it's dynein or kinesin, promoting the formation of the tubule. We think it's kinesin, but still a work in progress. So one of the things, and now I'm looking at the MBoC paper, that we realize when we look at our FIPSIN data set is that there is a clear interaction of the endoplasmic reticulum here on magenta with our tubules. Now, the ER can form, basically has different morphologies. So it's a very, very beautiful network in the cell, and it's composed of two different forms. So you have ER tubules that are very dynamic, as you could see in the arrows, and these flat cisterna in the arrowheads are called ER sheets that are less mobile. And what has been shown for a while, and pioneering work by Gia Volz and Jonathan Friedman, is that the mitochondria can actually, can actually constrict different organelles, in this case, the mitochondria. And that constriction can actually lead to membrane fission. So what I started to think is that maybe that ER interaction with the tubules may mediate or at least play a certain role in the fission of these tubules. And in fact, what we saw is on time-lapse microscopy is that in the constriction spot, which is the frame before the fission, there's this reduction in intensity of fluorescence, which is where the fission occurs. And we usually, about 70% of the time, we see an ER tubule sitting on it. And in fact, that is specific, because what you could do is to randomize the position of the ER by rotating it 90 degrees, and then compare the intensity of fluorescence in the original versus the rotation, and there is a decrease in intensity, meaning that the presence of the ER in that constriction site is not random. Now, we think that constriction of the ER promotes fission, but it's really hard to show that because you cannot knock down the ER. But what you could do is to modify the morphology of the ER. So in this case, what we did is to overexpress a protein called CLIM63. You could see in the MBoC cells, the ER is forming this beautiful, mostly tubular network. And when you overexpress CLIM63, that morphology changes and it's more prone to have very big sheets and less tubules. And when we compare the fission rate of our sorting tubules, what we see is a very strong reduction, which clearly suggests that the ER tubules are involved in fission. So basically, this is the model that we suggested on the paper. We know that this whole process, lethal, is driven by LOR2 and phosphoramine GIP4. And we know that at least partially, the endoplasmic reticulum, in a very similar way as the ER does in mitochondria and other organelles, constricts the tubules that promotes the fission. Now, there is still a lot to learn about this. For example, whether you have another GTPase or dynamines or things of that nature that also contributes to the fission, whether there's another organelle that might play a role with it. But at least we know for sure that they are playing a certain role to it, and there's still a lot more to know about the mechanism of this process and what does it mean for the cell and for the disease. So I would like to finish by thanking everyone at the NIH, especially Mark Cookson, my mentor, and everyone else that helped me through this process. And of course, everyone at MBoC for selecting my paper. As I said, it's a true honor, and it's been a really, really fun meeting so far. So now we'll gladly take your questions. Thank you very much.
Thank you so much. We have time for one or two short questions, and then I need to hand it back to Matt to wrap it up. But with that, I would like to say that if people have other questions for the speakers, I'm sure that all the speakers would welcome contact outside this venue to discuss further their science. So I see a hand up from Alejandro.
Okay.
Oh.
Hi, Luis. Awesome talk. So this goes a bit beyond your paper, I guess. So you said the ER constricts the lysosome. That probably is not physically the case, but then what would you think is actually the ER doing there? And if it would share common, or what would be the common ground for the ER going and chopping things into pieces?
What do you mean exactly on my process, or like
your process? And if it, so what are the ideas that you think the AR could be doing, and if it shares similarities with what it does at mitochondria or endosomes? Is it a common thing?
To your point, it is common in the sense that the ER it colocalizes in the spot prior to the fission frequently in many of those compartments.
Sorry, common, I mean like a common mechanism.
It is, that I don't know, because I guess it depends on what ER protein is contacting to what other, let's say, organelle protein, right? So I think, for example, for the mitochondria and for endosomes, actin plays a crucial role in it. What I've seen in my case, I don't see any actin puncta or colocalization to. So when I look at my tubules, actin is not there. So definitely actin does not seem to be, a point of bridging the ER with my tubules. So I think it may have some similarities, but I think it's also fairly different. For example, in the case of the mitochondria, the ER precedes DRP1 recruitment. I don't see DRP1 on my, of course, DRP1 is a mitochondrial protein, but I still check DRP1 and I don't see it recruited on the fusion spot on my tubules. I think there might be some shared things, but definitely I think it's more unique than other processes. And for what I've seen, when you change the morphology of the ER, you still see fission. So it's not like a complete blockage of it. So it might be helping a little bit, and might just be by interchanging lipids. That I mean, that I don't know.
I like that idea. It looks like something that you cannot delete lipids.
Yeah. So I did check VPS13C, which is a lipid transporter from the ER to lysosomes, and I don't see it colocalizing with my tubule. So it might, I don't, yeah, it's been difficult because I tried to do a split GFP for proteomics, but I could not get it to see it with DIP4 and Sec61B. So they may be too far away to actually get them. And so it's really hard to understand like what's in there if you cannot find like some sort of a split GFP or sort of a split apex or something like that you could look at.
Mm-hmm.
Okay, thank you. I think I need to turn it back over to Matt for a quick summary.
Yes, thank you, Claire, for hosting this webinar. Thank you and congratulations to all our speakers. Congratulations on your outstanding work. I wanted to just quickly draw your attention to the MBoC website. This is our most recent cover by Ofer Rogue and Janet Iwasa. And I also wanted to draw your attention to this little button here, which is our call for papers button. If you click on that button, you will see that we are now soliciting papers for a number of special issues featuring topic. I know it's very small, but I can just read a few of them: cell biology of bacteria and archaea, cell biology of the nervous system, biomolecular condensates, parasitology special issue, protein quality control, cell biology of the immune system, and all our long-standing quantitative cell biology issues. So I encourage you to go to the MBoC website, check out the beautiful cover, click on the call for papers button, and encourage you to support the ASCB and submit your latest, greatest work to MBoC. So with that, I think we need to draw the webinar to a close. Thank you to our panelists and our attendees. Congratulations again. And we hope to see you at the next MBoC webinar. So, all right, good morning, good afternoon, and good evening to everybody.